let's move on to the most, let's say, the practical that will be actually questioned in MCQs as well as part of the structure is the identification of reducing sugars. Okay, online students, you can hear me? Yes, audio, video, all clear? Okay, right. So reducing substances. What do you know about this concept of oxidation reduction? What do you know about this concept of oxidation and reduction? Do you know? Oxidation reactions, reduction reactions karada? No? Yeah. What do you mean by if something is getting oxidized? What do you mean if something is getting oxidized? Oxidized. It's what is happening? It's giving out electrons. Yes. It's releasing an electron. No? If something is getting reduced, it's taking an electron. No? It's taking an electron. It's taking an electron. But what is the word that we're using here? Is it oxidized or reduced? No, reducing is the word that we're using. So that means that is someone who's doing something, no? Right? He's not doing something to himself, he's doing something to someone else. Right? So reducing means he's reducing someone else, right? He's reducing someone else, right? What do you mean by reducing? That someone else is getting an electron, no? That someone else is getting an electron. That's what this reducing whatever the substance. So we call it a reducing agent. Okay. Reducing agent. Agent means like he's doing something. Reducing agent. So what is he doing? He's reducing someone else. Okay. And what about oxidizing agent? He's oxidizing someone else. Do you get it? Yes. He's oxidizing agent is oxidizing someone else. Okay. So so if there is no one to get reduced, no one will be oxidized. නිකං ආසාවට electrons දෙන්නේ නැහැ. Okay? If there isn't anyone asking for an electron, no one will give an electron. Right? ඔහේ නිකං ඉන්න ගමන් oxidize වෙන්නේ නැහැ කවුරුත්. Right? Get it? So always these two people has to be together. There should be someone asking for an electron to give an electron. There should be someone ready to be reduced for something to be oxidized. Okay? එහෙම නැහැ. අනිත් පැත්තට ගත්තොත් කවුරුත් reduce වෙන්නේ නැහැ, කවුරුත් electron එකක් දෙන්නේ නැත්තම්. Nobody is going to be reduced if no one is giving an electron. ඔය විදිහට you can say it both ways. Okay? So reducing agent is what? Is reducing someone else. Reducing someone else. That's what he's doing. He's reducing someone else. So how is he reducing someone else? By giving him an electron. Right? How do you reduce someone? By giving him an electron. So what is actually happening to the reducing agent? Yes, he's giving someone an electron. That means he's reducing someone else. So what is happening to the reducing agent? He's getting oxidized. So the reducing agent actually gets oxidized. Okay, reducing agent gets oxidized. Same thing for the oxidizing agent. He oxidizes someone else. Yes, oxidizes someone else. So that means what? He's taking an electron. Yes, that means he's what? The oxidizing agent is getting reduced. This is the concept you have to know before we move on to this whole reducing sugar concept. Okay, got it? Yeah. So how do sugars do this, right? And who is so reducing sugars, right? Reducing means what? He is getting oxidized, right? So the theory is there's a sugar. If he's reducing sugar, what is he going to do? He's going to give an electron. No, he's going to liberate an electron. Who is going to take this? In our experiments, we are using the ion copper 2 plus. Okay, we are taking the ion copper 2 plus, right? And what is going to happen? This copper 2 plus will become copper plus, right? So what is this? What has happened to copper 2 plus? Yes. So it's important that you get what is the theory behind it before you see it, right? So what is happened? What has happened to copper 2 plus? Is being reduced, reduced by the reducing sugar. Okay. Yeah. So this sugar is capable of liberating electrons so that this metal ion will be reduced. So when a metal ion usually undergoes a change in this number, what do you call this? An oxidation number, right? It undergoes a certain change in the aqueous solution, right? There's usually a color change. The color change that we can observe. Okay. Online students, audio all clear, right? It's not supported. What do you mean? Not a supported file. I sent it as a PDF, right? Okay, I'll try to solve that. Okay, right. Got this concept, right? So this is the concept behind reducing sugars, right? So in the last page of the tute, can you take this down? Not exactly this, what you understood, right? Basically, this is the concept that you need to know, right? What is going to happen, right? You can write down reducing sugars, just maybe in a pencil. So just you have a sort of idea, right? This is not a note, right? Just for your understanding, right? What the hell is a reducing sugar? You can refer this. Make your own small note, right? Then you understood it. So now you can write it in your own terms. And then I'll solve your tute problem. Hold on. Right. Moving on. Okay. So go to your this tute and let's see identifying reducing sugars in the lab. Right. So now the theory part. Okay. Reducing sugars such as what? What are the reducing sugars that we have learned? All monosaccharides, and out of the disaccharides, who are the only non-reducing sugar? Sucrose. Right. Sucrose the may prashna තියෙන්නේ sucrose විතරයි. So that's what we are going to do. We are going to do a special test for sucrose as a non-reducing sugar. Okay. So what are the reducing sugars? Highlight glucose, fructose, galactose, maltose, lactose. There's 5. Huh? Highlight them. Can be easily identified in the lab using the Benedict test. Okay. Benedict test එක let's try. So while we are preparing, if you go to any sort of graduate level, these tests are actually sort of your most fundamental tests, right? Even in our first and second years, we have a subject called biochemistry, right? For biochemistry, we have an exam called it's a bar exam, right? If you don't, it's called the first MBBS. If you don't pass it, you can't go on to the third year, right? And if you get repeated twice, they'll put you into the පල්ලෙහා batch, lower batch එකට දාන්න, right? එහෙම situation එකක් තියෙනවා. So in that, for the subject of biochemistry, we have a set of practicals that we have to do, right? Practicals tick කරලා we have to write a lab report, and there's an examiner also looking at what we are doing, right? And for those practice, so this is Benedict's solution. I'm not going to make it, right? There's a කඩේ ගන්න පුළුවන් Benedict solution නැති, right? This is basic alkaline copper sulfate, right? Copper 2 plus ions තියෙන, right? Basic solution. Basic means simple solution. It's also basic, right? As I was saying, for the biochemistry practicals, online students, you can see this table, right? Yes. What we are given to analyze for biochemistry is actually urine, right? Urine sample at this, right? So in urine, many things can be there. There can be glucose, right? There can be proteins. There's a thing called in your digestive system called bile. There can be bile, right? Many things can be there. So we are given, this is the Sudan 3, right? We'll come there. It's a simple reagent, right? This is HCl. So for the urine, the interesting thing is, where do you get the urine? For all experiments, there should be, ideally, there should be 2 samples that we do, right? There should be 2. One is the control, called the control experiment, and this is the test. Right? Usually, there has to be 2. Like at the same time, we have to do 2 like experiments. This is the control. So it means that is there to compare, right? So in a scientific experiment, you have to always compare. මේක තමයි මුකුත් නැති situation එක, මේක තමයි ඔන්න color change එක. You have to be able to show there has been some change, right? So as I was saying, in urine අපි test කරනවා glucose, protein, bile acids අරමේව තියෙනවාද, right? So do you have glucose and proteins in a healthy person's urine? If you go to the washroom and give me a sample, are you going to have glucose and proteins? Well, what sort of people have glucose in their urine? Urine paniraha. Yeah, it's in Ayurveda. Do you know how they identify? So what is the disease? Yeah. Diabetes, right? Ayurvedic in your traditional, yeah, diabetes. Online students, be interactive, right? In Ayurvedic practices, how do they identify diabetes? Did they have glucometers, blood darang test? No. How do they identify? They go, so they tell you to go and urinate near a tree, right? Okay, and if you attract ants to that urine, then they say you are a diabetic. That's sort of the theory. Why? Because in diabetes, there's a condition called nephropathy. So I'm going a bit out, right? Kidney damage, right? Nephropathy, diabetic nephropathy. Okay, diabetic nephropathy එක ආවම the kidneys are damaged, right? Imagine there's like the kidneys are supposed to be like a nice, very sensitive filter, but now the holes have become bigger. The kidneys are damaged. So big things such as glucose and proteins can leak from the kidneys into the urine. Okay, that's what's going to happen in diabetes. So a diabetic patient has glucose in their blood, sorry, in their urine. And what about proteins? There's another, so this nephropathy is going to lead to, over a long time, it is going to lead to a disease called CKD, chronic kidney disease. Yes, the CKD means, again, the kidneys are damaged. So as I told you, the holes of the filter is big now. Holes are big. So big things can come out, such as proteins are your big molecules, right? We learned last week, they are like big proteins, sorry, big molecules, globin proteins. Yes. So in a diabetic person or in a CKD person, there can be proteins as well. So where do we get that urine? We get that urine from the hospital, right? Diabetic clinic එකෙන් හරි kidney clinic එකෙන් හරි අපිට ඒ urine දෙන්න. So where do we get the control? Healthy urine ගන්න කොහෙන්ද? Batch එකේ කොල්ලොන්ගෙන් උදේට ගිහිල්ලා we have to urinate to a massive like 5 liter beaker එකක් තියලා තියෙනවා lab එකේ washroom එකේ. All the boys in the morning of the practical we have to go and donate urine. Right? So හොඳට වතුර බීලා එන්න කියලා. Right? So for the control we usually take the healthy urine from the batch. Right? And අස්සේ diabetes එකක් හිටියොත් ඒක හම්බෙන එක පවුද. Right? එහෙම නෑ. And sometimes මේ test කරන්න මෙච්චර urine හම්බෙන්නේ නැහැ. So in our batch there's about 190 no. හැමෝටම about 20 milliliters of urine දෙන්නවා. hospital එකම සුද්ධ කරලා ගෙන ඔක්කොම patient line ගත්තත් එච්චර නෑ. So what they are doing is normal urine වලට glucose උයි proteins උයි හැමෙන් දාලා දෙනවා මෙන්න test sample එක කියලා. Right? So that's how we do urine test, but we are not going to today do urine tests. For that we need gloves and like clean and no, we are just going to do some make some solutions and then we are going to do the test, right? So let's do the reducing sugar first. So what is the simplest reducing sugar that we can find in the anywhere? We can use glucose, right? We can use glucose. Okay. Then what is this? Kale bottle. This is honey, right? What is the sugar that we have in honey? You can remember nothing. Yeah. Fructose. Fructose tea. So අපි මේක කරමු glucose මුලින්ම කරලා then we let's try, okay? Positive වෙයිද දන්නේ නැහැ. මීපනේ සවුත් උනා. Oh, if it's a sugar, what would happen if it's honey made from sugar? ඒක non reducing වෙනවානේ. So it's going to be an issue. Okay? Online students, you are following, yes? What else do we need? This starch, sucrose, spirit lamp එකක් තියෙනවා. So I can't get a Bunsen burner here, right? So I had to bring a spirit lamp, right? And onion peel, let's do later, right? Don't ask where I got this stuff, okay? I didn't steal, but somehow I got them. That's the point. Okay. This is the biuret reagent, right? Yeah, you can better if you come. This row. This is the biuret reagent, right? So biuret, actually, according to your practical guide, you're supposed to make the biuret solution, right? But it's easy, but you can buy from the shops, right? You can buy the biuret reagent. You will know what are the composition of the biuret reagent, but for today, I'm going to use just the biuret reagent from the Kade. Okay. This is iodine, right? Iodine solution for what? What are we doing the iodine test for? Starch. Okay. So those are the things we are going to need. All right. Yeah. Okay. Starch, let's take later. So I need some help now. Okay. What did I say? This is a hands-on session. I need some help to go ahead with the experiments. First, let's make a glucose solution. Okay. Glucose solution. Can someone bring some water? Tapping with the tap, right? Right. So let's do the glucose test first for reducing sugars. Okay, then let's do the non-reducing, and let's see our sort of outcome. So yeah, there's already always when you do practical, it's always good to be organized, right? So usually you have to label, right? I already bought a labeled one. And what is this? Anybody know? This is a pipette filler, right? A high tech one. Okay, that we use to sort of suck reagents into. Thank you, Buddha. Right. So yeah, this is sodium bicarbonate. I think we're going to need that later. For mixing, I brought this here. Okay, so come back to your practical guide and read the method. So what is Benedict solution? We are going to do Benedict test now. It's a mixture of mainly copper sulfate and sodium hydroxide. Okay, so it's a alkyl. You didn't get. It's a what? It's a mixture of alkaline copper sulfate, right? You were here last week, right? Okay. Okay. This is the last week's sort of the day paper. Okay. Reducing sugars reduce soluble blue copper sulfate, copper 2 plus to insoluble brick red copper oxide, right? Cu, what is the compound that we are going to get actually? This Cu plus is actually very unstable. Yeah, vitamin aqueous solution, it is going to get, it's going to react with oxygen to produce Cu2O. This is the compound that we are going to get. This is actually brick red, right? Brick red. Sort of a, I mean, it's very unlikely that we'll go to this level, right? It's supposed to be a solid, right? That depends on how much electrons the sugar is giving and also how much copper you have in the Benedict, right? That's what it depends. So precipitate එක හැදෙන්නේ නැති වෙයි මේ සමහර විට, but we are definitely going to get a color change close to the red color. Okay, අර මුලින්ම බලාපොරොත්තු අඩු කරලා තිබ්බම වැඩි හොඳයි, right? So you might not get the precipitate, but we might get a color change. Okay, right. So let's make a glucose solution. Does it say 2 milliliters of reducing sugar into a test tube? Okay, at an equal, so let's make the solution first. Online students, you can see, right? So if it's better if you can make a very saturated solution until any more glucose can't be solved, right? I think it's close to being saturated, right? Can you see some glucose in the bottom, right? It's close to being saturated. At 30 parisimeter, let's add some more. Okay. Okay. So glucose appear 2 milliliters more. Now let's add a small amount. So the point is to add the equal amount of Benedict. Okay, got it? So then using our pipette, we need to take some Benedict solution. Right? This is, I mean, if you're doing this in front of an examiner, it's more nervous than this, right? කන්න වගේ බලන් ඉන්නවා. They are like අර label කරන එකේ ඉඳලාම solution එක හදන එකේ ඉඳලාම how you handle everything they are looking, right? And they are going to deduct marks if you set fire to anything or urine halagattut engapurama, right? So this is how you use a pipette filler. In your school lab, you might have like a rubber suctioning pipette filler එකක් තියෙයි ගොඩක් වෙලාවට, right? So we try to add the equal volume of Benedict. Online students, you can see, right? Bit too far away, right? Equal නෑනේ, තව shot එකක් විතර දාන්න වෙයිනේ. To be safe, let's add a bit more. It's like sometimes the Benedict solution can get like due to sunlight, the copper 2 plus can get sort of it can oxidize by its own, right? එහෙම reagent එක නිකන් එළියේ තියෙනකොට it can get weak, right? Weak වුණාම so the expected color changes are bit low, right? These are water, right? Yeah. Okay. I hope the ISM don't complain. I light fire inside, right? Okay. So what is this? Test tube holder, right? Nothing under there, right? So what does it say? Shake well. Did I shake well? No. Boil in a water bath. Why do we say boil in a water bath? That is to be safe, right? These are borosilicate test tubes, so they are not going to explode. Okay, so I can directly apply heat to them, but not continuously for a long time. So online students, you can see, right? Check by hands, right? So observe a color change. What's the color now? Blue, right? That is the color of what? Copper sulfate, right? Copper 2 plus ions is blue color, right? Online students, I hope you can see. I'll show you the camera, right? This is the blue color original. Okay. Can you see there's like slightly green color appearing now? What is the sequence of colors? Blue, green, yellow. Can you see? It's now green yellowish, right? Yellow, then it's going to be orange, right? Can you see? It's now green, yellowish, then orange, then finally it's supposed to be reddish, right? Brick red precipitate is very unlikely because of the strength of the Benedict, right? Can you see? It's now yellowish orange. Online students, can you see the color change? Right? Let's keep on heating it a little bit more. Now, again, what's the color? Orange, reddish, red dragon in it. Because of the AC, it's slow to heat. Orange again, other way. Let's see. I think that's the extent of the color change that we can observe, right? Sort of orange, red color එකට ආව නේද? Okay. Can you see? This is what? Glucose. Right? Glucose is what? It's a reducing sugar. Okay. Harida, what's next? What shall we do? Let's draw a diagram here. And here, initial blue color changes to brick red precipitate. Okay. Let's draw something. Right. This is what? Yeah. We usually say to take how much? Reducing sugar. So just say glucose, right? You don't need these values. This is the glucose solution. Next step, what did we add? Benedict's reagent was added. So now the test tube initial color was what? Blue, right? It turned blue, right? Then what did we do? We applied some heat. We applied some heat. What was the color change sequence? Blue, green, yellow, orange. Supposed to be at the end. There was supposed to be a precipitate, right? Yeah, there won't be a precipitate. That's why I'm මුලින්ම බලාපොරොත්තු අඩු කරේ, right? Brick red precipitate එකක් හැදෙනවා. What is the brick red precipitate පුතා? Cu2O, no? Yes. Draw this in the gap in the space given, right? Online students, any questions? Yes, check by ants. ගහ මුලට urinate කරන එක, right? Yes. Any questions about the practical? Is it sucrose? Sucrose. Oh, nice. So what do you say? Next, shall we do the honey experiment, fructose or non-reducing? Sucrose try කරනවද නැත්නම් fructose try කරනවා? Fructose ඔයගලන්ට කරන්න දෙන්න. Okay? So we add Benedict's, then we heat. There's a color change. Okay. Online students, any doubts? No. Video is okay, right? Then shall we do? Did I bring any beakers? I need to make sodium bicarbonate. Let's do non-reducing sugar experiment. Then let's do some other fun things. Okay, right? So, any questions about this? Highlight. What do you need to highlight in the first experiment? Initial blue changes to brick red precipitate. Highlight those. Uh, Equal volume of Benedict and what is the composition of Benedict, copper sulfate, sodium hydroxide. Those things highlight. Okay. Then moving on to a practical number 2, identifying non-reducing sugars in the lab. Right. 2 milliliters of non-reducing sugar into a test tube. Add a 1 milliliters of dilute HCl. Boil for 1 minute. Neutralize with sodium bicarbonate, hydrocarbonate. Check with pH paper. I didn't bring pH paper. Add an equal volume of Benedict solution. Shake well and boil in a water bath. Initial blue color changes to brick red precipitate. Right. Is this okay? Yes, this is okay, right? So now this is the same concept, but there's an additional step. Why are we going through an additional step? Because sucrose is a non-reducing sugar. So afterwards, can you see we are adding again Benedict's and heating, and we are getting the same color change, right? So we are doing something to sucrose. What are we doing? Acid එකක් දාලා heat කරනවා, right? Acid එකක් දාලා heat කරාම what is going to happen to sucrose? What do you think is going to happen to sucrose when we put an acid and heat? So sucrose H plus, and we are going to heat. No, we are H plus in HCl. We are going to add. Really? What are the monosaccharides or the monomers of sucrose? Yes, sucrose ඇතුලේ තියෙන මොනවාද? What are the two monomers, monosaccharides? Glucose and fructose, right? This is what's going to happen. How did we make sucrose? By releasing water, right? By a dehydration condensation reaction. We are going to do the opposite. We are going to digest, break down sucrose into glucose plus fructose. That's the purpose of doing this HCl and heating. Right? We are going to digest it. We are going to add some water. So what's going to happen? The sucrose is going to break down. So what about these two? Are they reducing or non-reducing? They are reducing sugars, no? Glucose and fructose are reducing sugars. So once we have done this, broken down the sucrose, we are left with these two. These two, we can do the Benedict's test, and it will give the normal Benedict color change. This is what we are going to do. Very simple. That's the theory. Okay. So, which sucrose do I use? Which? Right. This is our glucose, right? Like a patle now. So let's make a sucrose solution. Sucrose takes a while to dissolve in cold water, right? Let's keep it to get dissolved, right? So sucrose we added. Then what should we have in hand? Because we need what? Sodium bicarbonate. This is sodium bicarbonate. Why do you need sodium bicarbonate? Chemistry pantiyak kunani. Right? Why do we need sodium bicarbonate? To after adding HCl and heating, we need to neutralize it. Right? Acidic solution එකේ Benedict වැඩ ගන්න නැහැ. Right? Because Benedict works best in alkaline medium. So we after adding HCl and heating, we need to neutralize. Right? So let's make a sodium bicarbonate solution. Yeah, dissolve වෙලා. I think it's saturated, no? යට සීනි තියෙනවනේ. Sodium bicarbonate. Let's add a little bit more. We don't need him again, right? So this is your glucose, right? Upper balloon. Yeah, එතන නතර වෙලා, right? Orange, reddish color එක is stopped, right? There's no precipitate, right? So that's realistically the color change, right? Benedict is old, I think. Power එක අඩු වෙලා. So let's take another test tube to do our sucrose label, right? What do they say? About 2 milliliters of sucrose, right? Okay. Then what do we need to do? We need to add an HCl. So I'm going to remove this filler. We are going to use it for Benedictine, right? Let's take a separate one for the acid. Okay. You have to be very careful because if you suck the acid into the pipette filler, the right? So this is our dilute HCl, not that dilute, right? In the chutak ඇති. Any question? No. Sucrose, what do we add? We add the HCl. Okay. HCl added. What do we need to do next? What the right? So we need to heat it, right? To do what? We need to break down the sucrose into these monosaccharides so that we can do the reducing sugar test. Can I proceed here? Right. How do you say? Boil for 1 minute. So one of the biggest risks of doing experiments for girls is අපේ lab එකට යන්න දෙන්නේ with without tying your hair, right? Hair is very flammable, right? We don't use these sort of spirit lamps in the lab. No, we use what? Bunsen burners. Bunsen burners. There's a very hot, like a, and if you accidentally open the tap, well, there can be a big flame, right? If your hair catches on fire, there's no way to put it out, right? Hair is very flammable. Have you seen hair burning? Or you can smell it. Keratin burning is very pungent. No? Sorry, සඳහන් යන දෙන්නේ practical sort of, right? So big risk catching fire. It's boiling, no? Boil it on. Okay. Just keep it for a while. And what do we have to do next? We had to add some sodium bicarbonate to neutralize, right? මේකෙන් චුට්ටක් දාන්න. My students following, right? In the ගිහිල්ලද? See, that's the reaction, right? The acid is being neutralized, right? දන්නවනේ acid base reaction එක. Even I forgot that. It's a reaction. Right? So what's next? What do we have to do? Right? Where are we in the process? We have completed the breakdown of sucrose. Now we have to do the normal, the usual procedure that we know, right? මොකද්ද අපි ගන්නවා? Add Benedict. No? Benedict නම් ගමකට දාන්න වෙනවා because the solution is a bit too large. No, yeah. Otherwise, we won't be able to observe the color change. There's still some acidity, right? Can you see? Because it's reacting with the alkaline Benedict. So what does that mean? We need to add excess Benedict දාන්න වෙනවා නේද? Add a few more. It's the initial color now. After adding Benedict, blue color. No? Hopefully, මේකේ මෙයාටත් වඩා color change එක it's going to be bit low, right? Because what did we have to do? We had to break down sucrose, and the Benedict also reacted somewhat. No, acid එක්ක reacted. Let's see, right? If we get the required color change. So what are we doing here now again? We are doing non-reducing sugar test for sucrose. Always hold whatever you are doing away from your face, right? Basic. safety things, right? Push kala uturla out, you have to protect your face. Cross wasn't concentrated enough, maybe. No color change. No? I think the acid was a bit too much. Slight reduction in blue color is there. No? Slight reduction in blue color. Yes. Can you see? Several things could have happened, right? My sodium bicarbonate was not powerful enough to neutralize the acid. Benedict, can you see? The blue color has reduced somewhat, right? That means there is some reducing sugar action. Can you see? A slight brownish color, no? Reddish orangish වගේ color එකක් ආව නේද ලාවට. Let's try it again. You can try it again. I'll give you, if you want, you can come and do in the break even. Can you see online students? So this is realistically what you can expect, right? Orange color, right? Several things was not done properly, but I added a bit too much acid, right? So to neutralize that acid, I had to add too much bicarbonate. Not too much, I think too little, right? Acidity slightly remained, so that acid reacted with the Benedict. You saw, right? Because why? Benedict is an alkaline solution, though that might have reduced the power of the Benedict. Okay, that is likely. But can you see slight color change? We can, in the break time or even after the class, you can try, right? If you are interested, you can try by yourself, right? Benedict but it's not mine. Okay, but don't ask where I borrowed them from. Okay. Benedict, you have to keep it closed, right? It's going to react with the atmospheric oxygen. Okay. Clear? Online students. Shape එකේ orange තියෙනවා නැත්නම් මගේ නම්බු ඉවරයි, right? Yes. All right. So let's write this down. How can we modify this? How can we modify this reaction? How can we modify this experiment? Here, what's the step? HCl. That means H plus, and we are heating, no? H plus heating. What is the initial solution that we are taking? What's the initial solution that we took? Right? Sucrose solution, right? Sucrose solution. We added HCl, H plus H in HCl, right? Then we heated. Before that, before adding Benedict's, what else did we do? We added some sodium bicarbonate to neutralize. Then we added Benedict's reagent, so the solution became blue. Then we heated it again. Color change. May hari wage nathuruna ne orange wage vela. It also stopped. Okay, quickly take this down. Quickly. There's a precipitate. May glucose එක යට. Can you see? It's 2T precipitate. Yeah. You can you see? There's a small precipitate. What is that? Cu2O. Yeah. Can you see? 2T එකක් තියෙනවා. Right. So you have to trust your reagents. නැත්තම් වැඩි නොන්ඩි. Right. Online students, please take this down. Draw the diagram. Questions. Okay. Sodium hydrocarbonate is same as bicarbonate, NaHCO3. How sucrose is broken down? Yes. So your sucrose is broken down to glucose and fructose. Right? So then glucose and fructose are what? Reducing sugars. Then we can do the normal reducing. Sodium hydrocarbonate, yes. So it's sodium bicarbonate, right? Again, why we add excess of Benedict reagent is to make sure that we get the color change. Okay? Because who is our main I mean, let's say, who is the reacting species? Cu2 plus. Cu2 plus in Benedict. So you have to make sure there's enough Cu2 plus to get the color change we need. Right? Excessive Benedict, the underone. Okay? When we use dilute HCl, no, it's yes, it is necessary to neutralize. Why is it necessary to neutralize HCl? Because Benedict is a alkaline solution, and for the reaction to happen, it's preferred that this reaction happens in alkaline medium, right? Vedipur acid tibbot, it's going to affect the final color change. So that's what happened to me, right? Tikak acidity remain on because my dilute HCl is not that dilute. Wholesale kala gain වෙනවා එහෙම වුණොත් කැන්ටි මේ තියෙන්නේ coconut oil just to do the Sudan 3, right? It's a very Montessori experiment, right? Shall we move on? Any questions? Online students, any questions? Yeah. Percent blue highlight can highlight Benedict, right? And so you can use like some colors for the color changes, yellow, green. Okay. Moving on.